All attendees are in listen-only mode.
Good day to everyone joining us and welcome to today's X-Talks webinar. Today's talk is entitled, What is the Future of Cancer Clinical Trials? My name is Andrew Jordan and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes and this presentation includes a Q&A session with our speakers. This webinar is designed to be interactive and webinars work best when you are involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen, and if you require any assistance, please contact me at any time by sending a message using that chat panel. And at this time, all participants are in a listen-only mode, and please note that this event will be recorded and made available for future download. And at this point, I would like to thank IDDI, who helped develop the content for this presentation. International Drug Development Institute, or IDDI, is an expert center in biostatistical and integrated e-clinical services for pharmaceutical and biotechnology companies in several disease areas, including oncology and ophthalmology. IDDI optimizes the clinical development of drugs, biologics, and devices thanks to proven statistical expertise and operational excellence. Founded in 1991, IDDI has offices in Belgium, Boston, Massachusetts, Raleigh, North Carolina, and San Francisco, California. And now, I would like to introduce our first speaker for today's event, Dr. Everardo Saad. IDDI's senior medical expert, has over 15 years of experience in medical oncology and clinical trial designs. He graduated in medicine and trained in internal medicine in Sao Paulo and did his fellowship in medical oncology at the University of Texas MD Anderson Cancer Center in Houston. After practicing for several years, he shifted his professional career towards education and research in medical oncology and has a special interest in clinical trial methodology, the assessment of endpoints, and the development of novel therapies for cancer patients. Our second speaker for today, Dr. Jay Herson, is a collaborator of IDDI and formed Applied Logic Associates, or ALA, in Houston in 1983. ALA grew to be a biostatistical data management CRO with 50 employees when it sold to Westat in 2001. Jay joined the adjunct faculty in biostatistics at Johns Hopkins in 2004. His interests are interim analysis in clinical trials, data monitoring committees, and statistical and regulatory issues. He chaired the first known data monitoring committee in the pharmaceutical industry in 1988. He is the author of numerous papers on statistical and clinical trial methodology, and in 2009 authored the book Data and Safety Monitoring Committees in Clinical Trials. And now, without further ado, I would like to hand the mic over to Dr. Everardo Saad. You can begin your presentation when ready.
Thanks a lot, Andrew. Hello to all of you. On behalf of all of us from IDDI, I'd like to welcome you to our webinar. Jay Herson and I will try to provide some tentative answers to the question posed as the title of this webinar. And in order for Jay to discuss some possible futures, I will mostly set the stage for him by discussing the most relevant past and present features of cancer clinical trials, which we believe will be important drivers of future changes. So let me start by providing a very quick summary of the history of medical oncology. And I believe this can be very quickly summarized as follows. More than 100 years ago, Dr. Paul Ehrlich, who was actually working with tissue staining, postulated that some magic bullet would exist in the future that would allow for specific therapy for many disease types, not only cancer. And not very long after that, the biological effects of chemotherapy were discovered, and then the first designer drugs were developed. I show you here the molecular structures of 5-FU and tamoxifen as examples. And then came the era of clinical trials, initially for hematologic malignancies and then later for solid tumors. So important milestones were the cure of some types of lymphomas and leukemias. And then, of course, all the story of the adjuvant therapy of breast cancer, which is really very important in the history of medical oncology. In parallel to that, of course, molecular biology became increasingly important, and this gave rise in the late 90s to what we call nowadays targeted therapy. And then the new drugs like monoclonal antibodies and the tyrosine kinase inhibitors came to be. I will talk more about that in a few slides. Then with high throughput technologies and many advances in the laboratory, personalized oncology became a reality, and this is currently the cornerstone of medical oncology. I will talk about that more. So I think if we were to have a snapshot picture of the past and the present of medical oncology, lung cancer is a good example. If we look on the left-hand side of the slide and looking for the recent past, we see situations such as this, where chemotherapy was used mostly in unselected populations and in search for relatively small treatment effects. And very often, like in this case, no effect at all could be found, even though 4 chemotherapy regimens were compared. There was no difference between them in time to progression or overall survival. More recently, oncologists have become somewhat spoiled by looking at survival curves, or in this case, progression-free survival curves, with very dramatic treatment effects, such as what we see here on the right-hand side. And now we're talking about targeted therapy that's being used for selected populations in search or at least finding larger treatment effects. And this is really the way to go in terms of improving treatments for our patients. So what has changed? Obviously, there is an improved knowledge of cancer biology, and this has allowed for better drugs. Most of them are safer because these drugs target very specific genetic or epigenetic abnormalities found in the various tumor types, and also because we have been able to find biomarkers that have predictive power, and they allow for selecting populations that are enriched for the right target when that is successful. Unfortunately, that's not always the case, but when it is the case, we see the dramatic improvements that I have just shown you. So this has been called personalized oncology or precision oncology, if you will. Different people use different terminologies here. But what has been proposed is that the older approach of classifying tumors from a histological point of view and then treating such tumors with targeted therapies, sometimes finding low response rates in a few patients, will be replaced by another framework in which genomic pathology will play an important role, will be the focus of the assessment. So regardless of the tissue of origin, what's important is to look for specific signaling pathways that are abnormal in different tumor types, and then treat patients with targeted therapy, hopefully with the aim of achieving larger response rates or clinical benefit, however that is ascertained. So this is personalized oncology, and it owes a lot to targeted therapy, as I said. If we look at recent approvals by the US FDA and look at the blue bars, these are the number of new approvals for agents over the past 10 years or so, and we see that most approvals have indeed been targeted therapies. Immunotherapy is already represented here, and we know that this is very likely to improve in the future, or to increase rather, and so targeted therapy and immunotherapy are progressively substituting chemotherapy as the main forms of systemic therapy in oncology. This is a very interesting paper in which the authors provide very convincing arguments for why the efficiency of drug development is decreasing. So if you look over the years at the number of drugs that you can develop by spending $1 billion, and this is inflation-adjusted dollars, so it's in current terms, it's clear that whereas 50 years ago you could develop more than 10 drugs with this amount of money, nowadays you can barely develop one single drug. So the decrease is somewhat predictable, and the authors have humorously called this type of behavior Eroom's law. As you may know, Moore's law, which is used in computer science and engineering, predicts that computer power will increase every 2 years or so, will actually double every 2 years or so. And then this is the opposite of Eroom's law, in the sense that the efficiency of drug development will decrease in a predictable manner. So the authors took the word Moore and just spelt it backward to create Eroom's law. And then they go on to try to see whether clinical trials are to blame in this decreased efficiency. And so this picture is from the same paper. And if you look at the steps that lead to clinical trials, in other words, everything that goes from target identification to all the preclinical studies, and then to clinical trials and commercialization, they believe that in the phases of development that come before clinical trials, Moore's law is actually at play, in the sense that there has been improvements in efficiency in terms of identifying the potential targets and drugs. And the problem really seems to be here. So the bottleneck seems to be the fact that there have been comparatively small changes in the success of molecules entering clinical trials over the past 50 years. So in a way, clinical trials are to blame. So there is clearly a need for change in clinical trials, not only based on that paper that I just showed you, but on the opinion of many organizations and investigators. And even the FDA has proposed that the applied sciences needed for medical product development have not kept pace with the advances in the basic sciences. And this is the type of evidence that is presented for that. For example, the investment required for one successful drug launch has increased tremendously over the years. So if we compare between 1995 and 2000, and then 2000 and 2002, there has been an increase of approximately 60% in terms of how much money is required, and the FDA has identified what they call the critical path as likely to blame, because the money that's needed for discovery has not changed a lot. What has really changed is the money that's needed for the preclinical and the clinical phases of development. So they have named this the critical path, and they have launched the Critical Path Initiative, which is really the FDA's strategy to drive innovation in the process of development of drugs and other medical products. And the FDA has partnered with initially with Duke University and now with more than 60 organizations around the world, in order to try and really transform the whole clinical trials enterprise. And so this is called the Clinical Trials Transformation Initiative. And their website shows very interesting problems that they have identified and the proposed solutions to these problems. So this is likely to bring important changes for the near future. Other pieces that I would like to show you as possible drivers of the changes that Jay will talk about are these. There is, of course, an increasing amount of time that is required for completion of clinical trials. Again, this is just one of the possible views of this problem, but it's clear that between, for example, 1999 and 2002, and then 2003 and 2006, there has been a nearly 2/3 increase in the time required for some of the milestones in clinical trials. For example, for when the protocol is ready to the last patient, last visit, or to database lock, the time has increased tremendously. Another piece of this puzzle is the fact that the clinical trials enterprise is more and more a globalized endeavor. So whether you look at the number of centers or the number of investigators, it's clear that Western Europe and the rest of the world are playing increasingly important roles to the detriment of the role of the US alone as a country. Now, we could talk forever about the technological changes that will likely shape the future. I only show you here three components of these technological changes. One is the decreasing cost of sequencing the human genome. If we were to follow the cost that is predicted by Moore's law, it would have this kind of behavior, but clearly some disruptive lab technologies came about some years ago that made costs go down, and we're coming to a point where hopefully one genome will be sequenced by approximately $1,000 or $2,000, and this will be important for oncology in the future. I'm not going to talk a lot about nanotechnology. Jay will make some comments about that, but it's likely to be a player in the future as well. And for medical oncologists, there is a great appeal in what's called liquid biopsies, in the sense that you can obtain information about tumor biology in bodily fluids such as blood. So whether you look at circulating tumor cells or circulating DNA or RNA, this is very appealing because you can have a look at what's happening over time and not rely on information that was obtained like 1 or 2 years before some kind of decision has to be made for patients. Now, all this technology will, of course, increase the complexity of the decisions that need to be made. It's well known that the human brain can only deal with a few data points at a given time, 4 or 5 types of information. So historically, oncologists have looked at, for example, patient age, histological type, TNM stage, 1 or 2 markers, and that was enough for making treatment decisions. Whereas as time goes by, we increase our capacity to know about other aspects of tumor biology, including structural genetics by looking at SNPs, mutations, and other things like that. Also, the expression of genes by looking at RNA and then proteins. So the more we know, the more complex this will become, and very likely in the future, computer algorithms will be required for treatment decisions in the clinic as well as in clinical trials. How can we manage such complexity? There are many ways. Probably I just highlight here at least 3 types of approach. One of them I would call technological, which is the use of bioinformatics to deal with so-called big data. I will say a few words about that. Another approach I would call social, in the sense that we can harness the so-called wisdom of the crowd to help to ask for help from the community in order to save problems which are common to everyone. And then scientific approaches such as systems biology is one of the examples. So rather than looking at specific signaling pathways or genetic changes, we have to look at the whole picture and try to see systems properties that can be modified or understood at this higher level of approach. So it's also well known that the life cycle of knowledge is progressively becoming shorter, and we have to deal with that in clinical practice and in clinical trials as well. In terms of trials, we believe that adaptive trial designs will become more and more common towards the future. We talk about biomarker-driven trials, either umbrella or basket trials or other types, and these are already a reality, and they will probably become more common in the future. Another issue is the need to take a look at the data while the study is being conducted in an organized and structured fashion. So central statistical monitoring is an attempt to control the quality of the data, and we believe that this type of approach will become increasingly more important, and perhaps the best example in that regard is the CluePoints software. Now, information and communications technology are already important in healthcare and in clinical trials, and they will likely become more important as everyone knows. So they are useful for raising awareness and for disseminating results of clinical trials. They help to recruit and to for the retention of patients. They allow for real-time assessment of biological samples and other physiologic parameters. They allow for capturing, monitoring, and analyzing data, of course. They provide logistic improvements. One example is the mobile trials. Jay will say a few words, but these are situations where trials do not have to have fixed centers, and investigators become investigators when they are community physicians that have patients who qualify for clinical trials. So information and communications technology will be the way to link with those physicians and make them become investigators in so-called mobile trials. And then finally, electronic health records, which are already very important, will likely become a source of data to plan clinical trials. As many of you may know, the American Society of Clinical Oncology has just launched this initiative called CancerLink, which is really a very interesting health information technology platform that is promising to revolutionize the way we treat people with cancer, because this will allow clinicians and researchers to share their data, their clinical data about patients, and this will likely reveal important aspects of tumor biology that will be tested in clinical trials. So this is likely to be another source of data for planning clinical trials. And then my last slide, I wanted to say a few words about at least what I think is a trend for increased transparency in clinical research, with many initiatives historically with all the regulation, but then with things such as the consort family of guidelines. It's just one of the examples. Then clinical trial registration, which is also important for transparency, and public availability of databases is becoming a reality. There are many initiatives in that regard. So some sponsors have made their the data available after some clinical trials are completed. So this will likely be very important for generating hypotheses and testing some hypotheses in a retrospective fashion. Hopefully this will expedite the development of new clinical trials. And finally, I think there will be increased patient engagement in research, in particular in healthcare in general. I think patients should be more and more involved, even in the design of clinical trials in the future. So now it's my pleasure to pass the podium to our staff futurist, Jay Herson. Jay will talk to us about the next 10 years of clinical trials in oncology.
Well, hello everybody. I hope you can all hear me. Those of you who I have spoken to before, welcome back. And hello to all of my new friends out there. Let me give an overview of my talk. First, I'm going to talk about what is the role of foresight. What is it? What can it do and what can't it do as we plan for the next 10 years? What are the drivers of the future? And I'll explain what I mean by that. And what are the issues related to the drivers? And then what will oncology clinical trials look like in the year 2025? And what will remain the same? Now, the role of foresight, is it to predict what will happen? Well, we can never predict what will happen. There are all kinds of wild cards and things that will happen that we can't even conceive of. Is it to project from what is happening today into the future? Well, that's part of it, but it can't be all of that, because if you go back to 2005, everybody was talking about adaptive designs beyond just sample size re-estimation. Several conferences that year, many papers. So somebody could have said, okay, I will predict the future for 2015. All trials will be adaptive, will be adding arms, dropping arms, changing hypotheses through the trial. Well, has that actually happened? No. So we have to be careful about projection. We have to be careful that we're not just listing what we hope will happen. We have to realize back in 2005, there may have been speeches, legislation, and popular movies and novels, and someone would say, well, with all of this that I see, by the year 2015, we will be in a post-racist society. So you may disagree, but I don't think, unfortunately, that has happened. So we have to be careful that our predictions or our foresight is not what we hope will happen, because not everybody sees the world, or not everybody sees a gain in what we want to happen. But we can create scenarios of what is likely to happen, taking a lot of factors into account. And if we can create several scenarios, we are in a position to create a desirable future. What has to happen for this scenario to come true? If it gets off course, how do we get it back on course? These are the ways foresight is generally used. In this talk, I can't go through all of this. It's highly complex, but I will describe a plausible future for clinical trials in 2025. I'll be concentrating on pivotal phase 3 oncology trials, and mainly on the design and analysis of these trials. Now, what are the drivers? Well, certainly clinical technology, statistical methods, big data and big medicine, regulation, globalization, the cost of development and the cost of treatment for drugs that are approved, and what are the roles of generics and biosimilars. I'm going to cover each of these drivers in a moment. But first, the drivers offer suggestions of how the future might be shaped, but the future is driven mostly by the market, and trials will evolve in the direction that sponsors consider to be faster, cheaper, and most likely to result in positive trials. And we can't blame sponsors for doing that. So we always have to think of these three as we're trying to project what is likely to happen and what will not get very far. So now to go over the drivers, clinical technology, certainly we're going to be learning more and more about biomarkers, but every time we investigate a biomarker, we see that is not quite enough. Now, pathways, and I'll describe what I mean by pathways in the next slide, but they will interact with immunotherapy, immunogenic chemotherapy rather, and combinations of immunotherapy with targeted molecules. And there will be real-time data collection and safety response. A lot of this will come through some nanotechnology that we should see in 10 years, where nanoparticles in drugs can actually be sending signals about safety to the internet to the physician and the sponsor. So we won't have to wait for a data monitoring committee to have a meeting every 6 months to know if there is a problem. Now, in statistical methods, we have basket designs. Patients with different organ sites, but the same genetic alteration or mutation. So I'm going to call a pathway a genetic alteration or a mutation are enrolled. So it doesn't matter what the organ site is, but the patient should have the same genetic alteration, and different drugs that are supposed to address those will get into this basket design. And we will have uniform response criteria across organ sites by 2025, and I will explain that more. Of course, there's going to be enrichment. The more we learn, the more we can put patients on the trial, any trial that are more likely to respond. Now, one hears a lot about SMART trials, which seek to create dynamic treatment regimens. What that means is we start a trial and something happens. There is a toxicity. The protocol says, okay, when the patient gets this kind of toxicity, we now randomize them to either reduce dose or not reduce dose, or something like that, or another drug. But anyway, for different things that we can think of in the trial that could happen, we randomize a patient progresses. We can now randomize them to one or two different strategies. So at the end of the trial, you have all of these branches coming down, and pick the one that is best and do a confirmatory trial with it. That is the idea of SMART trials. Now, perhaps SMART trials can rescue us from intent to treat and overall survival. At least it can do that. Intent to treat, we've lived with it for 40 years. It is wonderful. It makes statistical sense, but it doesn't answer the question that physicians and patients have about the treatment. It only says that if we adopt a policy of starting patients this way or starting another way, this is what happens. Well, think of if we're doing SMART trials, different things happen, and we can randomize patients to different sub-strategies. When that happens, then intent to treat is more branches and more we can learn from it. Overall survival makes a lot of sense to the regulators, but the problem with overall survival is when the patients go off study because they progress, there are all kinds of different drugs they go on, and we're measuring survival, and sometimes there's crossover allowed in the protocol, and we don't really get to measure overall survival. All of the rescues that take place are make sense, and we want to continue them. But can we continue with overall survival as the primary endpoint if we're doing that? Big data, electronic medical records, pooled. Insurance companies have data, FDA has data, Walgreens and Boots have just merged. They have all of these electronic prescriptions. The UK has all of the data in Her Majesty's Health Service. All of these can be pooled, not all of them, but in different agreements, and we can learn from that. Cancer Link is an ambitious attempt by ASCO, just getting started, where every ASCO member, wherever they're practicing, will enter all of these data on their patients and then the outcomes, and then another physician gets the patient of that type and at least gets to see what the experience is with different treatments. They're not clinical trials, but they can suggest ideas for clinical trials. Big medicine. Oncologists are members of large organizations today. They're not in solo practices. And as a result, we can get people organized better to do clinical trials. Now, the Institute of Medicine in the U.S. wrote a paper that Everardo spoke about, a clinical trial enterprise. And what that means is any physician using Cancer Link, once he or she enters the data at diagnosis of their patient, a screen will pop up all clinical trials that patient is now eligible for. And if the patient consents, that physician will now be an investigator on that trial. And this is a more pragmatic way of doing trials. It won't necessarily be a university physician, and indeed this may be the only patient that physician puts on the trial. Regulation. There is inertia in regulation. There's skepticism on the part of regulators of all of the new ideas and how will they work. There's conflicting advice, 1 year to the next from regulators. Even a sponsor goes into a regulator. They go in February, they hear one thing, they come back in October, they hear something else. The ICH and the different guidances put out by FDA and the EMA, do they really tell us that much? I mean, everyone is waiting. When will the date come? When will the guidance come out, at least in draft form? When will it come? When will the next step of the ICH come? And then what happens? It says, write down your assumptions carefully. Justify your assumptions. Write your hypotheses. This really doesn't help us. It's really the community ourselves that have to come up with what seems right to us and press forward. Companion diagnostics with biomarkers, we have to have companion diagnostics. Well, they go through their own regulatory pathway separate from the drugs. And indeed, clinical trials are active using a companion diagnostic that still has not been approved. Not approved in every country, sometimes not approved in any country yet. How do we deal with that? What if it's not approved ultimately when the trial is over? And again, I have mentioned intent to treat, overall survival. We need some improvement there to go along with the improvements in medical technology and statistical methods. Now, globalization. Can non-Western countries follow complex protocols, use companion diagnostics and biomarkers? In this year, I'm working on a trial where one person, a sponsor, told me the Ukraine has technicians that can create and work on nuclear bombs, but they don't have a technician that can read the biomarker for a breast cancer trial. Will that develop? Are patient characteristics needed for the personalization distributed it evenly around the globe. We're going to learn this, but Big Pharma needs a global rollout of any new drug. That's what they're set up for. That's what their business model wants. Let me take an extreme. We come up with a really important patient characteristic that goes well with a biomarker, but those patients are located only in the state of Indiana in the United States. Is that drug ever going to come to market? That's an extreme example, but I think you can see what I mean. Now, cost of development. The cost of conducting trials may eventually need smaller sample sizes due to enrichment, but the need for companion diagnostics and other high tech will still make them costly. The cost of finding patients with the required characteristics. When we have the companion diagnostics, we may find only 20% of the patients we screen will qualify for the trial. This costs money. False positives will cut down our power, and false negatives mean that we have to increase our sample size. Both of those errors that they will make are costing us something. Now, mergers of pharma companies to reach critical mass for the development at this cost is at an end due to antitrust laws. We can't have just one pharmaceutical company worldwide. And most of the countries see eye to eye about antitrust. So pharma companies, we're going to see them working together in a way that I'll describe in a moment, partnering for development. Now, basket trials, again, patients are screened for molecular abnormalities and then assigned to treatment contributed by a pharma company. Pharma companies will be in what I call a pay-to-play situation, in huge screening basket trial organized by a nonprofit. So a nonprofit says, okay, for this molecular abnormality, we start in September. It's going to cost you, Mr. Big Pharma, so much to get into the game if you want to contribute your drug, and they'll all be then in parallel. And there are early signals about basket trials, and I think Everardo mentioned both of these. The NCI just announced a match. The molecular analysis for therapy choice, it's going to be run by ECOG Akron, and ASCO, the TAPER study, actually a registry, targeted agent and profiling utilization registry. ASCO actually getting involved and running a registry and running a clinical trial with the money they are now raising through the Conquer Cancer Foundation. This is all new and an early signal of what I think we're going to see down the line. All of this that we get out of basket trials will enable us to repurpose marketed drugs. There'll be an initial screening for new drugs and a repurposing for drugs already on the market. Now, the cost of treatment. After approval, treatment is expected to be expensive due to the smaller markets, due to personalization, and the biomarker testing with its false positives and negatives. Is coverage for this level of cost sustainable? Desirable? Again, I'll take this up again. The role of generics and biosimilars. It's not clear that biosimilars will lower cost because it is expensive to do a biosimilar trial, and we're still not sure what criteria the FDA will, and other regulators will ultimately accept. Will they give up on overall survival? Already we're hearing in the breast cancer trials, okay, do your trial, an adjuvant trial, but you better include pertuzumab. It would be unethical not to. Well, pertuzumab is very expensive. And then who knows, when the adjuvant trial is over, they may say, now do a neoadjuvant trial. So physicians are comfortable also with generics and approved drugs, likely to become biosimilars. So who knows that we will need new drugs? That is, we may be repurposing and using the drugs like docetaxel that have been around, and we don't need the new drugs. And then the ideas for improved use and personalization will come from the large databases. New ideas will come from smart trials and basket trials. Nonprofits will fund trials of generics and biosimilars, and those would be, as an example, the NCI, the MRC, Wiki foundations organized by Pathways. The year is now 2025. Development will be aimed at precision medicine. Pay-to-play basket trials will predominate. Global will not be a problem. Everyone will be up to speed by then. Databases and basket trials will guide large, simple, pragmatic trials of approved drugs. Bayesian methods will be allowed and encouraged here. Phase 1 and 2 will be merged if they haven't been already. We're not going to be doing 3+3 designs or CRM. We're going to be doing that together with the early phases that we find even in basket trials. Now, central statistical monitoring is big today. It's just starting out, but it may play a smaller role if data are coming direct from the patient into the clinical trial database, because so much of the quality of data is lost with the recopying and the re-entry of data, which may be eliminated. 2025, best overall response by increasingly sophisticated RECIST criteria will replace PFS and OS. The market will demand that. We already have early signals. The Cleopatra and the Neosphere adjuvant trials in breast cancer, they are using PCR and CPCR. I won't go into those definitions here. Pathologic complete response, I think most of you know that. But anyway, the post-approval submissions on cost effectiveness. Now, cost effectiveness will take overall survival into account. I mean, we just want to be sure that patients don't respond and the next month they all die. Cost effectiveness can be achieved through Bitcoin accounts. Patients can begin spend all of your expenses with Bitcoin accounts, and we can track those indirect costs, which are so hard to do today. Pharma companies will develop their own companion diagnostics. They're not going to wait for some other small company. They're going to either develop themselves, or they'll purchase licensing, or purchase smaller companies that are developing them. Development for small markets will be done by foundations created by wiki communities of patients and their families. Multinational pharma companies, wiki communities, and market forces will shape regulation. The multinational pharma companies are in every possible country. They are more important than the FDA or the EMA. Multinational public CROs will evolve into research pharma companies themselves. And today's pharma companies will be more marketing companies. They've got the sales force and the support staff worldwide, but already CROs are doing more in the development process. There'll be more contract manufacturing. No more waiting for a company says, I have to do an interim analysis. I need to know for this biologic, do I have to build a new factory? There will be a factory out there already approved that is doing manufacturing for several companies. And the smaller CROs will not be left out. They'll be running the basket trials under contract with the nonprofits. Still more to develop for precision medicine. Only about 30% of cancers will be treated in this way in 2025. Smart trials, yes, to save overall survival endpoint from the crossover fever. Yes, to ease the inferential burden of intent to treat. No to creating dynamic treatment regimes that will be abandoned by 2020, if not already, as a bridge to nowhere. Too many branches. What remains the same in 2025? Doxorubicin and cyclophosphamide, etc., in developing countries and for the non-insured in the USA. Randomization followed by increasingly sophisticated data analysis, Bayesian methods, causal inference. We see the early signals today. There'll be skeptical regulators, and ASCO will meet in Chicago May 30th to June 2nd of that year. Mark your calendars, and you can see the future becoming the present. Thank you.
Great. Well, thank you so much for that, Dr. Herson. Very insightful. Also, thank you to Dr. Saad for his portion of the presentation. And at this point, I would like to open up to the audience for their questions or comments. Right now, you can send in those questions using the chat window for this, which is the Q&A portion of the webinar. And I've already received a number of questions here, so let's get started. Our first question here. This audience member writes, it seems there are reasons to think phase 3 clinical trials will be larger than they are now as a result of multiple scientific questions, smart trials, and a more pragmatic design, as in comparative effectiveness research, but also that they will be smaller due to enrichment and improving treatment effects. Which way do you think the trial size will actually go? So to either of you.
Everardo, do you want to start?
I guess you can tackle that, Jay. Will you?
Yeah, sure. We are going to have in the future large trials and small trials. The trials will be large because in many cases we always have to have a control group, and there may be small differences because we have sophisticated active controls. So I think we will have smaller trials. Of course, the basket trials are small. They are learning trials. So we will have them, and I don't think we're going to get away from large trials. I think also we're going to have pragmatic trials, large, simple trials. When we get into the repurposing of drugs, which will come from ideas from, as we said, the databases and whatever, and smart trials. For those drugs already approved, we will have large, what Sir Richard Peto called with his work with the thrombolytics several decades ago, the large simple trials. And we will need, they can be large because there will be virtually no safety data that will need to be collected since all the drugs are approved. But that would be a way of answering a question that comes right out of a database. And with large, simple trials, we're not in a position to do that today. So I would not say that trials are going to be smaller in 2025.
All right. Well, thank you for that. Moving on to another question here. How do you think, if the trials become more pragmatic and will try to imitate real-life situations, will it hamper the justification of the testing of hypotheses and or their registration of a new drug? Or is it better to be more conservative in the terms of the clinical trial design and enrollment criteria?
Do you want to answer that, Everardo? Because I'm not sure I have that question on my screen.
Well, yeah, it's quite complex. I think it touches on issues also from the previous question. Andrew, can you repeat? I guess the question has more than one component.
Absolutely, yeah. It's a very long question here. They're basically asking if trials become more pragmatic and try to imitate real-life situations, will it hamper the justification of testing the hypothesis and or the registration of the new drug, or is it better to be more conservative in terms of the clinical trial design and enrollment criteria?
Yeah, I guess what made me think of the previous question is that probably the answer is, I think that there will be a role for both situations. I don't think we'll move away from testing hypotheses, as Jay mentioned in one of his slides. We believe randomization will still be around, so even in trials that are not randomized, some kind of inferential thinking will be required. So I think testing hypothesis will still be there, but of course I think there will be a role for changing some of the designs and the issue of enrollment criteria. So I think there will be trials where will be more pragmatic, imitating real-life situations. That actually happens already when you think about cooperative group trials and trials that test strategies rather than drugs, purely speaking. But there will also be a role for testing new drugs or combinations, and this will have to rely on hypothesis testing, comparing with conventional standard of care or controls, if you will. Jay, do you have a different take at that? This?
No, I agree with you, Everardo. I think that.
Okay, all right.
All right, well, thank you for that. Moving on to another question. This audience member writes, I understand that results of clinical trials with cancer vaccines have provided surprising results. For example, effects on overall survival, but not on DFS or PFS. Can you explain this? Do we understand that rationale? Can you indicate the lessons to draw for future trials with immunotherapy in oncology?
Jay, can I give it a try?
Yes, go ahead.
Yeah, I guess no one actually knows. I wish I knew the answer to this question, because there's clearly something in immunotherapy that makes one think that there is this discrepancy between overall survival and some of the intermediate endpoints, for example, PFS. And there have been examples. One of them that comes to mind is in the adjuvant setting with Provenge, autologous vaccine. And I think there are other situations where there have been gains in OS and no gains in PFS or DFS. I guess at this point no one knows if this is something biologic, so to speak, if there is an explanation that is particular to immunotherapy, or if it's just the play of chance, or whether there is some kind of biased assessment of the intermediate or surrogate endpoints. I think no one knows the answer at this point. But clearly, immunotherapy is something that will be more and more important, especially in combination with other modalities. And I think there will be efforts to understand how immunotherapy influences the current endpoints, or at least how we see the endpoints and how we evaluate treatment effects, and I think that remains to be discovered.
Yeah, and I would add that in prostate cancer, we have found that I think it was the SHARP trial, with the most recent one, where again the PFS was not predictive of OS, and there are various reasons for that. But I think the important thing is to be aware in which diseases, for which types of drugs, is it predictive and is it not predictive. And with more factors, as we learn more about biomarkers, companion diagnostics to get patients into the trial, there's going to be more and more of this disconnect. That's why I think pathologic complete response is likely to be the basis of approval, and with follow-up studies in the future, because these long-term large sample sizes, yes, but long-term follow-up for these endpoints is not, it often raises more questions than answers them, and it can be a heartbreaker for a sponsor that has spent so much money on these trials.
All right, excellent. I think we can squeeze in one last question here. HIV and AIDS has been cited as a model for cancer research, and rapid detection of resistance in combinations of antiretroviral cocktails when resistance is detected. What can we learn from HIV and AIDS research to apply to cancer trials?
Jay, do you have experience?
One thing, I guess, from the Concord study, we learned that we have to do follow-up for one thing. But I think but that's, now I'm going back 3 decades now. I mean, more in the present. We are learning, of course, how to do trials in the third world, but I think we're learning the role of patient groups, wiki communities, and the like, the role that they can play in shaping design, in actually running trials, financing trials, and that kind of thing. They've been very active from the beginning, and because of that, we now have groups of patients with the breast cancer, even the triple negative foundation, prostate cancer, and others that are the wiki communities of tomorrow that are going to actually be running these trials and demanding more sensible endpoints, looking at data from themselves, from the large databases to make recommendations.
All right. Well, thank you very much for that. We have reached the end of the question and answer portion of this webinar. If we weren't able to address your question today, the team at IDDI may be able to follow up with you after the webinar. If you do have any further questions, however, you can direct them to the email address showing on your screen right now for Dominique Groussard. Her email is dominique.groussard@iddi.com. And thank you to everyone for participating in today's conference. You'll be receiving a follow-up email with access to the recorded archive for this event, and a survey window will be popping up on your screen. Your participation is appreciated as it'll help us improve our further webinars. Now, please join us in thanking our speakers for today, Dr. Everard Osad and Dr. Jay Herson. We hope that you found this conference informative. Have a great day, everyone.